Hey learners, welcome to BioCell Academy. Today we are diving into one of the most mind-blowing concepts in biology, that is stem cells. Imagine a cell that holds the potential to become anything, a brain cell, a heart cell or a skin cell. That's the magic of a stem cell. And understanding them is a key to unlocking the secrets of development, disease and regeneration. So let's unravel this incredible story from a single cell to the complexity of an entire organism. Our journey begins with the stem cell itself. Stem cell, the terminology was given by Theodore Boveri and Valentin Hacker. Our key properties of stem cells. Self-renewal. This is crucial. Stem cells can divide to produce more stem cells, maintaining their population. High proliferation. They can divide many times, creating a large number of cells. High potency. This refers to their ability to differentiate into various cell types. The higher the potency, the more types of cells they can become. Stem cells resides in stem cell niche. That means these are specific microenvironments in the body that support and regulate stem cells. They are specialized nurseries. Now let's talk about the types of stem cells, categorized by their potency, essentially how many different cell types they can become. Totipotent, the most powerful. These can differentiate into any cell type, including extraembryonic tissue like the placenta. Think of the zygote, the very first cell formed after fertilization. That's totipotent. Pluripotent. These can differentiate into any cell type of the germ layers, ectoderm, mesoderm and endoderm, that can make up the embryo. However, they cannot form extraembryonic tissues. Example, inner cell mass. Multipotent. These cells have more restricted potential. They can generate multiple cell types, but typically within a specific lineage or tissue. For example, mesenchyme stem cells and hematopoietic stem cell. Unipotent. As the name suggests, these can generate only one cell type. Example, spermatogonia and oogonia. Stem cells don't just divide randomly. Their division patterns are critical for maintaining balance and generating diversity. There are two division types, symmetrical and asymmetrical. In symmetrical, a stem cell divides to produce either two identical daughter stem cell or two identical differentiated stem cell. This is how the stem cell pool expands. In asymmetrical division type, this is where it gets really interesting. A stem cell divides to produce one daughter stem cell to maintain the pool and one differentiated cell. This is a fundamental mechanism for generating diversity while preserving the stem cell population. How does an uncommitted stem cell become a specialized heart cell or a neuron? Well, it's not a sudden jump. It's a gradual journey involving two steps, specification and determination. Specification is the first step where a cell becomes committed to a specific fate. But this commitment is still reversible. Think of it as a soft commitment. If you put that specified cell into a different environment, it might change its mind and develop into something else. There are two types, conditional specification and autonomous specification. Conditional specification is also known as regulative development. Here, the cell's fate is determined by its interaction with neighboring cells and the environment. It's about cues from outside. If you remove cells, the remaining cells can often compensate and regulate their development to form a complete organism. This is common in vertebrates and some invertebrates. Autonomous specification, also called as mosaic development. In this case, the cell's fate is determined by factors within the cell itself, often unevenly distributed in the cytoplasm. It's like the cell already has its instruction pre-programmed. If you remove cell, the remaining cells cannot compensate and a part of the organism will be missing, leading to a mosaic of defects. The next critical step is determination. This is when the cell's commitment becomes irreversible. Once a cell is determined, even if you move to a completely different part of the embryo, it will still develop into its determined fate. It's logged in. This is a crucial checkpoint in a development, ensuring that cells faithfully carry out their specialized roles. Finally, after determination comes differentiation. This is the process where the cell undergoes morphological and functional changes to become a specialized cell type. It acquires its unique shape, expresses specific genes, and performs its specific function. Whether it's contracting as a muscle cell, transmitting signal as a neuron, or secreting hormones as a gland cells. This is the culmination of the stem cells journey. Pluripotency in embryonic stem cells is maintained by a specific set of transcription factors like SOX2, OCT34, and NanoG gene. Scientists like Yamanaka showed that by introducing just a few genes, often referred to as OSKM factors for OCT4, SOX2, KLF4, and CMYC, we can reprogram differentiated somatic cell into induced pluripotency stem cells or iPSC. This has revolutionized regenerative medicine and our understanding of cellular identity. From an uncommitted stem cell to a fully functional specialized cell, the journey of differentiation is a testament to the intricate dance of genes, proteins, and cellular interactions. Understanding stem cell isn't just academic, it holds immense promise for treating disease, regenerating damaged tissues, and even understanding aging. The field of regenerative medicine is buzzing with possibilities, all thanks to these incredible cells. If you found this exploration fascinating, hit that like button, subscribe to BioCell Academy for more biological deep dives, and let me know in the comments what other cell biology topics you would like to explore next, or even biology topics in general. And hey, if you want a PDF of this detailed notes to help with your studies, the link is in the description below. Make sure you are following BioCell Academy on both YouTube and Instagram for more amazing biology content. Stay curious and keep learning. See you in the next video. Bye bye.